Hello friends, welcome to Allied Classes. Today in this video we are going to talk about some historical background of DNA and Watson and Crick DNA model. As if we are talking about the Watson and Crick DNA model, so the DNA model was proposed by the Watson and Crick. We'll write here the topic name DNA. DNA is a nucleic acid. There were the two types of nucleic acid, deoxyribose nucleic acid and ribose nucleic acid. So we will write here DNA is a nucleic acid. And who said that DNA is a nucleic acid? So the Friedrich Miescher reported that the DNA is a nucleic acid. So we will write here Friedrich Miescher reported that the DNA is a nucleic acid. Okay. Now, as DNA is a nucleic acid, it is a acidic substance. DNA is a acidic substance. Now, where DNA is present? The question arises where DNA is present. So, each and every cell of our body consists of equal amount of DNA. DNA is a master molecule of our body. Now, the term, as we know that the structure of DNA is given by Watson and Crick. So, we will write here structure of DNA is given by Watson and Crick, and they got Nobel Prize in 1962 for the DNA model. Now, the term DNA was given by Zancris. So, the term given by Zancris. Now, we are going to see about the DNA. DNA is a polymer of nucleotide. Means many monomers are formed to form a polynucleotide chain. So, we will write here DNA is a polymer of nucleotide. But which nucleotide? Deoxyribonucleotide. Deoxyribonucleotide. There were the two types of ribose sugar, pentose sugar. There were the two types of pentose sugar, ribose sugar and deoxyribose sugar. In case of DNA, there will be deoxyribose sugar, and in case of RNA, there will be ribose sugar. So, the DNA is made up of three main components. Which are they? Pentose sugar. Pentose sugar are of two types. As we said that deoxyribose sugar and ribose sugar. Nitrogen bases. Nitrogenous bases. In case of DNA, there are ATCG, means adenine, cytosine, thymine and guanine. And in case of RNA, the nitrogenous bases are adenine, uracil, cytosine and guanine. So, in case of DNA, if we are talking about DNA, so there will be adenine, thymine, guanine and cytosine. And the third more important component is phosphate group or the phosphoric acid. Phosphate group, or we can call it as a phosphoric acid. Now we are going to see that how this nucleotide is formed by these three components. So we will draw the structure of pentose sugar and how the nitrogen base get attached to the pentose sugar and how the phosphate group get attached at the pentose sugar. So we will draw here the structure of pentose sugar. Here the oxygen, 4 carbon atom. You can see here the 4 carbon atom, 1, 2, 3, 4. And the 5th carbon is attached at the arm side. So, this is a pentose sugar. If you are talking about deoxyribose sugar, then it will be here double H, and here the functional group is attached. And if we are talking about the ribose sugar, then there will be at second carbon and third carbon there will be the functional group. So we are talking about the deoxyribose sugar. So at third carbon there will be functional group, at second carbon there will be not a functional group present. Now here you can see at the first carbon the nitrogen base is going to be attached and forms a glycosidic bond here. Glycosidic bond. At first carbon the nitrogen base get attached to form a glycosidic bond. The bond between carbon and nitrogen base is of glycosidic bond. If we talk about purines, if the nitrogen base is purine, then it is going to be form the glycosidic bond at the 9th carbon. And if we talked about pyrimidine, then it will be form the glycosidic bond at the first carbon. Okay, so there were the two cases on the basis of nitrogenous base. If there is a purine on the place of nitrogen base, then it will be form the bond of glycosidic at the 9th carbon, and if it is pyrimidine, then it will form the glycosidic bond at the first carbon. Now we will see how this phosphate group or the phosphoric acid get attached on the 5th carbon of pentose sugar. So here you can see the phosphate group is going to be attached. Phosphate group is going to be attached. We know that the structure of phosphate group is like this. Here you can see OH, OH and OH. When this one functional group get attached to the 5th carbon, then it will be formed of water molecule, and here one bond is going to be formed between phosphate group and the 5th carbon of pentose sugar. This bond is known as phosphodiester bond. Phosphodiester bond. So this is a structure of nucleotide. Here you can see it is a structure of nucleotide, or you can say it as a deoxyribonucleotide. If we are talking about deoxyribose sugar, then it is a deoxyribonucleotide. Deoxyribonucleotide. Okay. Now we are going to see the Watson and Crick DNA model. Now we will write here about the Watson and Crick DNA model. Watson and Crick DNA model. According to this Watson and Crick, the DNA model is a double helical structure. dsDNA means it having a helical structure with 2 strands. So we will write here DNA is a double helical with 2 strands. DNA have 2 strands. Now this each strand is made up of chain of nucleotides. As you can see, there commonly we find a double strand DNA and a single strand RNA. But there were some exceptional cases where single strand DNA is found and double strand RNA is found. So we will write here first. It is a dsDNA. Okay. Means each strand of DNA is made up of chain of nucleotide. These two strands of DNA are antiparallel to each other. Two strands of DNA is antiparallel. Means from 5' end to 3' end and 3' end to 5' end. Now you know that in case of DNA there is one purine and one pyrimidine. According to chargafs law, one purine and one pyrimidine is present, means the equal amount of purines and pyrimidines are present in case of DNA. So you can say we can say it as a purines pairs with pyrimidine, means adenine pairs with thymine and thymine pairs with adenine, vice versa. Cytosine pairs with guanine and guanine pairs with cytosine. So we will write here purines pairs with pyrimidine, pairs with pyrimidine, means adenine pairs with thymine forms 2 hydrogen bond. Here you can see it forms 2 hydrogen bond, means in adenine and thymine there were the 2 positions where hydrogen bonds are formed. Hydrogen bond. Cytosine pairs with guanine forms 3 hydrogen bonds. Here you can see 3 hydrogen bonds. It means that there were the 3 positions where the hydrogen bonds are formed in case of cytosine and guanine. Okay. Now we will see the structure of DNA proposed by Watson and Crick. Before that we have to see how these nucleotides are formed to form a long chain of polynucleotides to form a DNA. So first of all, if you want to understand the structure of Watson and Crick DNA model, we have to understand how these nucleotides are get attached to each other to form a DNA model. So we will draw here the structure of nucleotide. Here you can see the pentose sugar, here another pentose sugar. Here we know at the first carbon the nitrogen base get attached, so we will attach here adenine, here cytosine. Here at the 5th carbon we know that the phosphate group is attached, so we will attach here the phosphate group. Now here the 5th carbon is free, so we will write here as a 5th prime end. Here the 3rd carbon is free, so we will write here 3rd prime end. Now we will draw here the anti-parallel strand as the DNA have. Here you can see one nitrogen base, here you can see the another nucleotide. Here we know that the phosphate group is attached. Here phosphate group is attached. Here adenine pairs with thymine. We know that one purine and one pyrimidine, and cytosine pairs with guanine. Here you can see the 3rd carbon is free, so we will write here 3rd prime, and here the 5th carbon is free, so we will write here 5th prime. So you can see the 5th prime to 3rd prime and 3rd prime to 5 prime. Here you can see this functional group is going to be formed bond with phosphate group, and this functional group is going to be formed a bond with this phosphate group. Now here the phosphodiester bond is formed between this nucleotide, and here phosphodiester bond is formed. Okay, and we know that in case of adenine and thymine there were the 2 positions where the hydrogen bonds are formed, so we will draw here 2 hydrogen bonds, and in case of cytosine and thymine there were the 3 positions where the hydrogen bonds are formed, so we will draw here 3 hydrogen bonds. Here you can see this is a hydrogen bond. So this is structure of nucleotide, how they are going to be attached and form a helical structure, and in brief detail we are going to see the Watson and Crick DNA model. Now here we will draw the structure of DNA given by Watson and Crick. So here you can see So this is the structure of DNA double helix. 3' end to 5' end, and here 5' end to 3' end. Here you can see, here the hydrogen bonds are formed. Here you can see the hydrogen bonds are formed. This is a double helical structure given by Watson and Crick. Okay. Now here you can see a shallow region, and here also you can see a shallow region, and here you can see a deep region. So this shallow region is known as a minor groove. Minor groove. And here deep region is known as a major groove. Major groove. We will write here minor groove. So this is a double helical structure. Now according to Watson and Crick, this one minor groove to another minor groove completes one 360-degree rotation. Means the distance between two successive minor groove or two successive major groove is of 36 degree. 360 degree, one turn. And this two successive major or two successive minor groove consists of 10 base pairs. Means one turn of 360 degree consists of 10 base pairs. If you want to see the angle between two successive base pairs, then the angle between two successive base pairs is 36 degree. And the distance between two successive base pairs is 0.34 nanometer, or if we talked about angstrom unit, then the distance between two successive base pairs is 3.4 angstrom unit. And we know that the distance between two minor groove or two major groove is 34 angstrom unit, or if we talked about nanometer, then it is 3.4 nanometer. So we will write here distance between two successive minor groove or major groove is 3.4 nanometer or 34 angstrom unit. Now this is about the DNA strand given by Watson and Crick, and the distance between two strands means the diameter of DNA is 20 angstrom unit. If we talked about nanometer, then it is 2 nanometer. The distance between two strands or the diameter of DNA is 2 nanometer. So this is the structure of DNA given by Watson and Crick. Next video we are going to talk about the types of DNA, that is aDNA, bDNA, cDNA, ddNA and zDNA. Well as some chargafs rules given for the DNA model, such that adenine will pair with thymine, cytosine will pair with guanine, forms 3 hydrogen bond, and when adenine pairs with thymine forms 2 hydrogen bonds. The rules given by the chargaf we are going to see in the next video. So thank you friends for watching my video.